Here we are, and we go next to the complement, right? So the complement, what you have to know, where is that? Where's the complement? Where are the complement proteins? Where are they located? Where are they? So they can access to every tissue. The blood. Okay, complement is in the blood. And principle of activation, what you have to understand, 3 types of stimuli, 3 pathways. Remember, classical, alternative, and lectin. What stimulates each pathway? Classical, antigen-antibody complex, lectin, mannose residues on the pathogen surface, and alternative, different proteins and carbohydrates, just various molecules on the surface of pathogens. Okay, and what you have to see that is that all those pathways, they actually they actually come to the same intermediate, the C3, which then causes 3 major outcomes: inflammation, phagocytosis, okay, and membrane attack complex formation. Okay? Understood? Right? If you have any questions, please ask me.
What is the membrane attack?
Remember that when those proteins assemble themselves in the membrane of the bacterial cell, essentially forming a hole, okay? And this hole allows water to enter the cell and cell lyses. It's like making...
So it perforates the bacteria.
It perforates the bacterial cell wall, yes. Cell wall and membrane, okay? So general understanding of adaptive immune system, what you have to realize here. So it is highly specific, okay? When we say highly specific, it's not like a relative term. Adaptive response will be against a single antigen, right? Effects of it is connected to the innate immune system via its potency to increase inflammatory responses, right? And stimulate complement activation via the antibody antigen complexes. Okay? Major properties: specific, and it forms memory. Okay, remember that. That's kind of 2 most important advantages. It's highly specific, and it forms immunological memory. The antigens, understand what can be and what cannot be an antigen. Big complex molecules can. Big, simple, or small molecules cannot. But you can turn small molecule into the antigen. The small molecules that can be turned into antigens, we call them haptenes. Okay? Remember, we stick haptene to a large carrier molecule like a protein, and the model organism can develop immune response to the haptene. So haptene will be an example in this case. So antigenic determinants are the fragments of antigens that immune system develops the response to. How many antigenic determinants a single molecule, single antigen can have? 1 word answer. How many antigenic molecules, how many antigenic determinants a single antigen can have? A lot. Okay. 2 words. Okay. A lot. Okay. More than 1. Can be 1. Well, sometimes it's 1, but the idea is that it's not limited by 1, right? Oh, yes. So you absolutely can expect the list of different molecules, okay, like a list, and at least we'll say which of these is not an antigen? Cannot be a complete antigen. Like a thyroid hormone, a DNA molecule, a polysaccharide molecule, and a protein. Thyroid hormone is too small. Or like amino acid, okay? Amino acid, protein, DNA, or polysaccharide. Complex polysaccharide. Amino acid can't, right? The idea of major histocompatibility complex, we'll come back to it when we review. Again, the function is to present antigens. There are two types. Know the pattern of expression, that type 1 is on all nucleated cells, type 2 is only on antigen-presenting cells, B cells and macrophages. Okay, cells of adaptive immune system. So what you have to understand about those, so we have T and B cells, right? That they start in bone marrow, then B cells go into the bone, mature in the bone marrow. T cells mature in thymus. Okay? You good in that? Difference between effector cells and memory cells. What are the effector cells? Yeah, the ones that actually make immune response happen. Memory cells,
They multiply quickly.
After second infection, second exposure. Okay, so they store information about previous exposure. VDJ recombination. That's how the whole variety of different receptors for antigens is achieved. All you have to know about VDJ recombination is, I had a question today, we also covered it in micro. The question today, do we have to know how many V genes, how many D genes? Hell no. You don't have to know these numbers. But you do have to understand you have V genes, D genes, and J genes. And by recombination, those genes, you can yield almost an endless variety of variable regions on antibodies or receptors. That makes sense? So this V, D, and J, these fragments, they form variable regions of antibodies and receptors on the cells. And they can recognize virtually any antigen. Maturation, well, you have to know about this. What I kind of should have said, thymus in bone marrow, okay? So this is for maturation. Positive and negative selection. Here's what I mean. Remember we talked about the positive and negative selection in the thymus, like positive selection, cells that can recognize MHC, that can produce immune response are selected, and during negative selection, cells that will recognize self-antigens will be killed. That's what I mean by positive and negative selection. Positive selection are immunocompetent. Negative selection, non-self-reactive. Does that make sense? Does that make sense? Okay, so understand the difference during positive and negative selection. Secondary lymphoid organs, we'll come back to it. It's here just to remind you that B&T lymphocytes, they enter the lymphatic system. Cool? Antigen-presenting cells. Dendritic cells, macrophages. You can count B cells as antigen-presenting cells too. Okay. What do? They present antigen. Yeah, that's about it. They present antigen. That's kind of it. Now, more specific about each branch of the immune system. Tell me if it approaches 15 minutes. Otherwise, we'll just stop 15 minutes. There will be a mismatch between audio and video. Branches of adaptive immune system. So humoral response. Which cells mediate it? B cells. Things that you have to know about them. Look, I almost gave you the answers here in the study guide already. They mature in the bone marrow. Clonal selection. What's clonal selection? That's when they respond to a specific antigen. Remember, there's so many clones of B cells due to VDJ recombination. So when B cells recognize a specific clone, the specific clone of B cells recognizes a particular antigen, that clone starts to expand. Clonal selection, clonal expansion. Result of the clonal expansion, B cells, what are effector cells for them? Plasma cells. Plasma cells produce antibodies. Okay? So understand that B cells per se do not make antibodies. They have to become plasma cells first. Okay? Now, primary secondary response. When you experience primary infection, what is the first antibody type that you produce? IgM, and then IgG. It's primary response. During secondary response, you have overwhelming amount of IgG. Does that make sense? Now, during the secondary response, what's the difference between primary and secondary? During secondary response, how is that different from primary in terms of the antibody amount? Amount of antibodies produced during secondary response. A lot more than the primary, and it is produced much faster. Why? Memory cells. I was expecting exactly this answer. 2 words. Memory cells. Okay. It's like, that joke, the non-traditional state mottos for West Virginia. The key to our obesity problem, 2 words, pepperoni rolls. For Ohio, by the way, happily swinging for more than half of the century. Swinging. It's a swing state. Active and passive, natural and artificial humoral responses. Expect, we're going to talk about this, they're going to pause. Expect the question that will give you a situation, like infection with blah, 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 is active artificial, active natural, passive artificial, passive natural. Got it? And you may expect opposite. Best example of passive artificial response is and answers, and you have to pick. So please understand, yes.
The only question I have is, what's the difference between active and passive again?
So in active response, great question. In active response, your immune system is actually exposed to the antigen, whether it's an infection or vaccine. In passive response, you passively transfer antibodies from a person with antibodies to a person without. Does that make sense? You see the difference? Okay, great. Another question. Oh, yeah, we're getting there, yeah.